We're going to finish up talking about meiosis today, and just to start again with the question, what would happen if haploid cells were not created? So the problem that meiosis solves for us is that meiosis produces these haploid cells of 23 chromosomes in the egg and the sperm, and that when they combine, the zygote has the correct number of chromosomes for a human, which would be 46 chromosomes. So if these haploid cells were not created, if we tried to take a 46 chromosomes egg cell and a 46 chromosome sperm cell, we'd end up with a zygote with 92 chromosomes, and that would not work properly as for a human. So wrapping this up, let's talk about how meiosis is different in males and females. The process of making gametes is called gametogenesis. Okay, so gameto referring to the gametes, the sperm and the egg, and genesis meaning the beginning. So in males, in the testes, they have cells that start out in a process to go through they start out and they go through a process of meiosis. So they have an interphase where the DNA doubles and homologous chromosomes separate and go into each new cell. This is the haploid stage. However, their sister chromatids are still duplicated chromosomes here. And those sister chromatids split in meiosis too, and the result would be 4 sperm cells. Now, these sperm cells are obviously not identical to the parent cell because they have just half the number of chromosomes, and we can assume that crossing over and independent assortment also occurred, which created these 4 unique sperm cells. Men are going to make sperm in the testes. They make them their entire lifetime and unlimited amounts. So if we go over into females, a woman in her ovaries has special cells called oocytes that are going to be the precursor of eggs and polar bodies. And so this goes through meiosis 1. Prior to that, in interphase, it's going to make a copy of DNA, and each cell is going to have homologous pairs are going to separate, and each cell is going to have half the information, except it's still going to be doubled. In that case, it goes through meiosis 2, sister chromatids separate, and you have 4 cells that have half the information. However, the one cell, the ovum or the egg cell, has a lot of the cytoplasm and organelles, and there's just an unequal cytoplasmic division. They all have the same nuclear amounts of information, but cytoplasmic division is unequal. And so we call these other 3 things polar bodies, and they secrete polar bodies. Women have a limited number of oocytes that will turn into a possible egg, and they release 1 of these eggs a month, and this happens in their ovaries. So you should practice listing maybe like 3 differences between male and female gametogenesis and 2 similarities. You can pause the video and check it out. And I'm going to flip the page and show you the answers. So if you haven't paused yet, you want to do that. And here are some possible suggestions of how students compared the 2. So pause it and look over your answers. Moving along, it's a good time to talk about these chromosomes, and that a scientist can take a picture of these chromosomes when they're in metaphase, once these chromosomes have condensed, and then a scientist can organize them using a computer to arrange them according to their size. And so you can see their largest to smallest, and the scientist pairs them up with a homologous chromosome. Okay, these are homologous chromosomes. They are not replicated. If they were replicated, they'd be attached here at a centromere. And we call the first of these chromosomes 1 to 22, we call those autosomes. And we call the last pair sex chromosomes. So take a look too down here. It explains how you get 1 from your mom, 1 from your dad. They have the same genes arranged in the same order. They just have slightly different DNA sequences. So in an egg cell, you're just going to have a karyotype would have half those chromosomes. In a sperm cell, you would have also half of the chromosomes, except in a sperm cell, the last chromosome is either going to be an X or a Y. It could carry one or the other. So a human karyotype is a picture of chromosomes arranged in homologous pairs and organized by size. Humans have 46 chromosomes. 2 of those are sex chromosomes. So an XX means that you would be genotypically a female, and XY is genotypically a male. The other 44 chromosomes, so 22 all the way back to 1, are known as autosomes. So what happens if meiosis doesn't happen properly? We call this failure to separate nondisjunction, which means not coming apart. So it could happen in meiosis 1, or it could happen in meiosis 2, that here the homologous chromosomes fail to separate. So now any future cells, 2 of them are going to have too many, the other 2 cells are going to have too few. Or if it happens in meiosis 2, nondisjunction may result in 2 of the cells being incorrect, but the other 2 would still be proper. So this is where random fertilization, which egg is fertilized or which sperm is fertilized, would potentially play the role in determining if this is going to produce a problem in the offspring. So here's a picture of a woman, and the woman is the one we're going to show the abnormal egg. It could be the male. A woman with an abnormal egg with 24 chromosomes, a woman with an abnormal egg with 22 chromosomes, 1 too few, 1 too many. If you combine those with normal sperm, this offspring has 47 chromosomes. We say that's trisomy. Some of the trisomy disorders are Klinefelter's, Down syndrome, Edwards syndrome. So you can Google those and check those out. Here we have 1 less chromosome than normal, and it's going to be called monosomy, so 1 less of mono. An example is Turner syndrome, and you can also certainly search for other monosomy disorders. Please make sure you go back and review the phases of meiosis, terms like crossing over, tetrad, what the overall outcome of meiosis is, and feel comfortable with the terms haploid and diploid.